In our today's lecture, we're going to talk about cells, the difference between pro and eukaryotic cells, different cellular structures, their function. But before we get there, we need to talk about how we study cells. We study cells using microscopy. There are two types of microscopy currently used: light microscopy and electron microscopy. Light microscopy obviously uses light, which can be refracted by the lens within the microscope. This refraction results in the image being inverted upside down and left to right. But in addition to that, microscope lenses increase the size of the image, enabling us to see the objects that are otherwise invisible to the naked eye. The microscope that we're going to work with in the class is called light because it uses light, compound because it has many objective lenses, binocular because it has two eyepieces, parfocal, meaning that it remains in the relative focus when the objective lenses are changed. Microscopes are characterized by three main features: the magnification, the resolution, and the contrast. Magnification reflects how much bigger the object looks and can be calculated by multiplying the magnification of the eyepieces by the magnification of the objective lenses. Resolution is the capacity of the microscope to distinguish fine details of the image. Resolution is limited by the wavelengths or the frequency of light, and the greater frequency gives a microscope the greater resolution. Finally, the contrast is how different in brightness are different parts of a microscopic image. The first microscope was developed by the Dutch merchant and amateur scientist Anthony van Leeuwenhoek. This microscope was later improved by the British scientist Robert Hooke, who was the first to observe the cellular structure of a cork, but unfortunately for him, he did not pay much of an attention to that discovery. Let's talk about the main parts of a microscope. As a scientist, you're going to look through the eyepieces to see the microscopic image. This image can have different sizes because there are several objective lenses directed at the specimen, which is placed on the stage. The microscopes that we're going to work with have a mechanical stage. It means that you can move that stage front and back, left and right, using mechanical stage knobs. The vertical position of the stage, and therefore focus, can be regulated by coarse and fine focus knobs. The larger knobs, the coarse focus knobs, and are used only in conjunction with the smallest magnification of the objective. In other words, with a scanning objective. Objective lenses are positioned on the revolving nosepiece, which allows to quickly change them. Since it's a light microscope, you have to have a source of light, illuminator. The brightness of illuminator is regulated by a rheostat. Condenser is the part of the microscope that focuses the light on the specimen, while the diaphragm regulates the amount of light that reaches the specimen and therefore regulates the resolution of the image. If the object is too small to be observed by the light microscopy, electron microscopes can be used. They use a beam of electrons instead of light. You can see the electron microscope here in the center. Electron microscopes have greater magnification and resolution compared to the light microscopes. On the image A, you can see a typical light microphotograph. And you can see that this unit represents the distance of 25 micrometers. The image B represents the scanning electron micrograph, and the similar sized unit now represents 1 micrometer. So you can see that it produces much greater magnification and much better resolution. Two main types of microscopy are transmission and scanning. Image 1 shows you the typical transmission electron microphotograph of Ebola virus. You can clearly see the inside surface, the inside structures of that virus. So if you need to see inside of the cell or inside of the virus, you would use transmission electron microscopy. Scanning electron microscopy gives you an image of the surface of the cell, but not the inside. So if you want a really high resolution, this is of course artificially colored, the image B is artificially colored. But if you want a high resolution image of the cell shape, like in case of this Campylobacter jejuni, you would use scanning electron microscope. So how do the cells appear? The original idea was that life is generated spontaneously. However, Italian scientist Francesco Redi delivered the first blow to this viewpoint. He placed pieces of meat in the open container, in a container that was sealed with a cork, and a container that was sealed with a gauze. Not surprisingly, flies landed on the meat in the open container, and eventually maggots appeared on the meat. In the cork-sealed container, not surprising at all, flies did not land on the meat and there were no maggots. However, in the container that was covered with gauze, flies landed on the gauze, not on the meat, and maggots appeared on the gauze. So even though air could reach the meat, meat was not in contact with any form of life. So Redi suggested that life, in fact, is not generated spontaneously and have to come from some other life. At the end of 19th century, Louis Pasteur pretty much put the final nail in the coffin of the spontaneous generation theory. He prepared 2 curved neck flasks. Both were thoroughly sterilized. That sterilization resulted in the formation of the water seal in the neck that preserved the sterility of the broth within the flask. On one of the flasks, the neck was broken, and this flask quickly demonstrated bacterial and fungal growth, unlike the one that remained sealed. After that, German scientists Schleiden, Schwann, Remak, and Virchow suggested the cell theory, proposing that cells only come from other cells, and when cell divides, it produces 2 identical daughter cells. It was before the meiosis was discovered. How different the sizes of the cells in different organisms are? Well, the smallest one are the viruses. Prokaryotic cells are larger than them. Unicellular eukaryotes usually are larger, and multicellular eukaryotes are largest. So which ones would require what type of microscopy? Well, multicellular eukaryotes usually are visible with a naked eye. Prokaryotic cells and unicellular eukaryotes in general require light microscopy for observation, while the viruses can be only seen in the electron microscope. Of course, there are exceptions from each of these rules, but we're going to focus on the general principles, not the exceptions. So what limits the size of the cell? Why can't we pretty much have a cell that is the size of the soccer ball? Well, the answer is nutrition. Prokaryotic cells are very small. which makes the diffusion of nutrients and wastes across the membrane very quick. Nutrients quickly get into the cell, while the wastes leave it. Eukaryotic cells are generally larger, so they had to develop various adaptations to increase the rate of the transport inside and outside of the cell. How does the diffusion limit the size of the cell? Well, for this, we need to look into the math of the cell. Let's say we have a hypothetical spherical cell. Its surface area is proportional to its radius squared. Its volume is proportional to its radius cubed. So when the radius of the cell increases, its surface area increases as the r squared, while its volume increases as the r cubed. In other words, the volume increases quicker than its surface. If you will write down a ratio of the surface area to its volume, you will see that as a cell increases in size, the ratio of surface area to volume decreases. Same concept can be applied to a cell that is shaped like a cube. If a cell is too large, the surface of that cell is not sufficient relatively to its volume to maintain the adequate rate of diffusion. To circumvent this, cells often increase their surface size by various types of foldings on the plasma membrane, or cells can become flat or thin and elongated. Eukaryotes also developed certain organelles that would compensate for the less efficient transport of the membrane, which we will discuss in our next lecture. Despite the fact that prokaryotes are very small, there are a couple of examples of the prokaryotic cells that are actually visible with the naked eye. The two largest bacteria, Thiomargarita magnifica and Thiomargarita namibiensis, can be so large that cell of Thiomargarita magnifica can be 9 mm long. You can see in image 1. And the cells of Thiomargarita namibiensis have a diameter of about 0.75 millimeters, which is greater than the diameter of the human hair. Now let's take a look at the structures that are found in cells, and specifically in prokaryotic cells. All cells, without exception, have a plasma membrane, cytoplasm, DNA, and ribosomes. In prokaryotic cells, which are unicellular organisms that don't have a nucleus or membrane-bound organelles, DNA exists in the form of a nucleoid. It's a circular DNA, a single circular DNA molecule that is bound to so-called nucleoid-associated proteins. The main function of nucleoid is to store and replicate genetic information. Nucleoid is submerged into cytoplasm, a gooey solution of proteins, carbohydrates, and lipids that fills up the cell. Ribosomes, found in prokaryotic cells, are responsible for the translation of mRNA, which results in the synthesis of proteins. They consist of two subunits, 30 and 50S, which are in turn composed of ribosomal RNA and proteins. And finally, every prokaryotic cell is surrounded by cell membrane. Cell membrane is a phospholipid bilayer. We talked about it in our previous lecture, which contains various transmembrane proteins that span the thickness of that bilayer, as well as peripheral proteins, can be seen here and here, which are located on either outside or inside surface of the membrane. The key function of the cell membrane is the transport inside and outside of the cell and the maintenance of the cell structure. In addition to these four structures that are found in every prokaryotic cell, many prokaryotic cells, but not all of them, have inclusions. Non-membrane structures that essentially are accumulations of various chemicals, shown here, which function as a storage for nutrients, metals, or whatever chemical cell needs. Some prokaryotic cells form endospores. Endospores are formed under harsh environmental conditions, with water, nutrients are absent, or toxic chemicals are present. Endospore allows the preservation of genetic information of bacterial cells for a long time and consists of, well, bacterial DNA that is surrounded by the coat made of carbohydrates, proteins, and glycoproteins. You can see an endospore here and a bunch of endospores stained green on this tiny little image on the right. Many bacterial cells have cell walls. Cell walls consist of peptidoglycans, basically chains of carbohydrates or glycan chains, in which molecules of N-acetylglucosamine and N-acetylmuramic acid are connected by glycosidic bonds. These glycan chains are linked together by peptide linkages that consist of amino acids. The main function of the cell wall is to maintain the cellular integrity, protect it against lysis, and control the transport of stuff in and out of the cell. In prokaryotes, especially in bacteria, cells can be divided into gram-positive and gram-negative based on the structure of the cell wall, and you can see on these images that predominantly the structure of peptide linkages is different between gram-positive and gram-negative bacteria. Finally, some cells have glycocalyx, which is the layer of carbohydrates outside of the cell wall. We want to put the order of the cellular structures. We start with cytoplasm that is surrounded by the cell membrane. Outside of the membrane is the wall, cell wall, and outside of the cell wall is glycocalyx. So, two types of glycocalyx that you must know are capsule and slime layer. Capsule is more rigid, slime layer is more loose. Capsule can protect cells against phagocytosis, destruction by other cells, especially cells of the immune system. And both the capsule and the slime layer can increase the adhesion of the cells to the surface. On the image on the right, especially if you enlarge it, you can see the capsule stain, where capsule is stained white. Many, but not all, prokaryotic cells have a number of external appendages. First of them is a flagellum, and cells may have multiple flagella. Flagellum is responsible for the cell movement. It consists of basal body, essentially a motor, that rotates a rod. Rod is attached to a hook which also rotates, and at the end of the hook is a filament. When filament rotates, it rotates like a propeller, and the cell is moving forward. Fimbriae are external appendages that look like a brush of proteins on the surface of the cell that can increase the cellular attachment to the surfaces. And finally, pili. Pili are long structures made out of proteins that can either facilitate attachment or enable cells to transfer genes between each other. Flagella can be arranged in 4 different ways. If there is one flagellum at the pole of the cell, we call this monotrichous arrangement. If there are 2 flagella on the opposite ends of the cell, we call it amphitrichous arrangement. There is a bunch of flagella on one end of the cell, we call it lophotrichous arrangement. And finally, flagella located all over the cell surface represent peritrichous arrangement. Now, eukaryotic cells differ from prokaryotes because they have a nucleus that is surrounded by two membranes, membrane-bound organelles, and, unlike prokaryotes, they have not one but several chromosomes that are not circular, like in bacteria, but linear. Organelles are essentially the cell compartments that have specific functions. So the cellular functions are compartmentalized in eukaryotes, which explains why eukaryotes have a more complex structure than prokaryotes. On these two images, you can see an animal cell on the left and the plant cell on the right. And next, we're going to talk about the main structures within the eukaryotic cells. First and foremost, of course, it is a plasma membrane. So plasma membrane consists of phospholipid bilayer with a bunch of proteins, transmembrane proteins, like these channels, and signaling proteins. We call them integral proteins, as well as peripheral proteins, here or here. These proteins are often connected to the filaments of the cytoskeleton, enabling cells to receive signals from the outside. The main function of that phospholipid bilayer is to control the passage of chemicals such as molecules, ions, water, oxygen, and wastes into the cell and out of the cell. In some cells, membranes are folded into projections called microvilli. These folds dramatically increase surface area and therefore increase the absorption across the membrane. The space between the plasma membrane and the envelope of the nucleus is called the cytoplasm. Cytoplasm contains all the cellular organelles which are suspended in the substance called cytosol. It's a gel-like solution of proteins, carbohydrates, and other organic compounds, and serves as the place for many metabolic reactions. The main component of cytosol, of course, is water, but the presence of proteins gives it a semi-solid, clot-like consistency. The center of eukaryotic cell is a nucleus. Nucleus is where DNA is. Nucleus directs the synthesis of ribosomes. It regulates the expression of genes and therefore the synthesis of proteins, and the nucleus is surrounded by a nuclear envelope. Nuclear envelope consists not of just one bilayer, but of two independent membranes, the inner and outer membranes or leaflets. Within the nuclear envelope, there are nuclear pores, openings that allow the passage of ions, molecules, and RNA in and out of the nucleus. Nucleus is filled by a nucleoplasm, which is similar to cytoplasm or cytosol in its chemical composition and is also a semi-solid fluid-like substance that maintains the nuclear shape. DNA in the nucleus is organized into chromosomes. In eukaryotes, chromosomes are linear and represent the hereditary material that carries the traits from parents to offspring. Every eukaryote has a specific number of chromosomes. For instance, humans have 46, fruit flies have 8. When the cell grows and during the life cycle, proteins are attached to the chromosomes and they make chromosomes look like unwound bunch of threads, shown here, called chromatin. Chromatin consists of both uncondensed, like this, and condensed, like this, chromosomes. Another structure found within the nucleus is the nucleolus. The function of the nucleolus is to assemble ribosomes by aggregating ribosomal RNA and ribosomal proteins. When ribosomal subunits are assembled, they are transported out of the nucleus, and it's important to notice that nucleolus isn't membrane bound. So what is a ribosome? Ribosome is a non-membrane bound structure that is responsible for the synthesis of proteins. Essentially, it is a massive complex of RNA and proteins which make up two subunits, the large and the small. Usually ribosomes exist as clusters. So for instance, if this is the mRNA molecule and ribosomes attach to it, they will attach to it like clusters that are called polyribosomes, and each ribosome in this cluster will produce a polypeptide chain. Within the cell, ribosomes can exist in the cytoplasm in the form of polyribosomes, can be attached to the plasma membrane, can be attached to the membrane of rough endoplasmic reticulum, or even to the nuclear envelope. If you remember our cookbook analogy, you can appreciate that when DNA is transcribed into messenger RNA within the nucleus, that messenger RNA is transported out of the nucleus via nuclear pores to be translated into a protein by the ribosomes. We just mentioned the term endoplasmic reticulum, which is a membrane of the endomembrane system of the cell. It's a group of organelles which synthesize, modify, package, and transport lipids and proteins. Endomembrane system consists of a nuclear envelope, lysosomes, vesicles, endoplasmic reticulum, Golgi apparatus, and plasma membrane. Endo means inside, and membrane means, well, membrane, so inside membranes of the cell. Endomembrane system does not include mitochondrial chloroplasts, which are also surrounded by membranes, but their function is completely different. The member of the endomembrane system that stands out is the endoplasmic reticulum, or ER. It's a series of interconnected sacs and tubules, which function is to synthesize and modify proteins and synthesize lipids. Terminologically, the space within the tubules and sacs is called a lumen. And the membrane of endoplasmic reticulum is essentially continuous with the cell nuclear envelope, which means there are no gaps between the two. Endoplasmic reticulum can come in two shapes, rough and smooth. Rough endoplasmic reticulum is called this way because of the ribosomes on the surface that faces cytoplasm. When ribosomes synthesize the proteins, these proteins end up in the lumen of the rough endoplasmic reticulum, where they undergo various modifications. These proteins can stay in the ER membrane or can be secreted by the vesicles that get released from the surface of the rough endoplasmic reticulum. Another important function of rough ER is to produce phospholipids for various cellular membranes. Unlike rough ER, smooth endoplasmic reticulum is responsible for the synthesis of carbohydrates, lipids, and steroid hormones. Since no proteins are involved, smooth ER does not have any ribosomes. Another important function of smooth endoplasmic reticulum is to detoxify various poisons and to store calcium ions, which play an important role in various intracellular processes. Near endoplasmic reticulum is Golgi apparatus, or Golgi body. It is a series of flat membranous sacs which sort, package, tag, and distribute lipids and proteins that were synthesized in the endoplasmic reticulum. The size of the Golgi body that is closer to the endoplasmic reticulum is called its cis phase, and the opposite side that faces cytoplasm is called trans phase. The vesicles that are formed in the endoplasmic reticulum transport substances from the endoplasmic reticulum to the cis phase of Golgi body, fuse with it, and empty its content into the lumen of the Golgi body. Proteins and lipids that were delivered by those vesicles travel through the Golgi body or Golgi apparatus and undergo various modifications. These modifications include, but aren't limited to, addition of the short carbohydrate chains, phosphorylation, addition of sulfhydryl group, maybe hydroxyl group. Then these modified proteins and lipids receive a tag, packaged into special secretory vesicles. The secretory vesicles leave the trans phase of Golgi and deliver their load to wherever this load is intended. In addition to its functions that we listed before, Golgi apparatus in plant cells synthesizes polysaccharides. Mitochondria, also known as the powerhouse of the cell. The main function of mitochondria is to produce ATP, adenosine triphosphate, the main energy-carrying molecule of the cell, which stores the energy in the cell in the short term. ATP is produced in the process of cellular respiration, which uses the chemical energy of glucose or lipids. Mitochondria carry out cellular respiration by oxidizing glucose with oxygen, producing carbon dioxide and water, which are essentially the wastes. Depending on the function of the tissue, cells may have, in that tissue may have many or very few mitochondria. For example, muscles that need a lot of energy for contraction are very rich in mitochondria. These organelles have the oval shape, and they are surrounded by two membranes, just like nucleus. Interestingly, these organelles have their own ribosomes and their own DNA, both looking like ribosomes and DNA from bacterial cells. This allowed Lynn Margulis to suggest that mitochondria are in fact endosymbionts. They used to be aerobic bacteria that were acquired, engulfed, by the proto-ancestral eukaryote. Structurally, the inner membrane of the mitochondria is folded into folds called cristae, and within the inner membrane is mitochondrial matrix, the place for many chemical reactions involved in the generation of ATP. The space between the inner and the outer membrane is called the intermembrane space. Other structures that can be found in eukaryotic cells include peroxisomes, vesicles, and vacuoles. Peroxisomes are fascinating organelles. It's a membrane-bound organelle that carries out reactions of oxidation of fatty acids and amino acids to generate energy. They can also detoxify many poisons, so in some way they are a weird mix between mitochondria and endoplasmic reticulum. Many oxidation reactions within the cell release hydrogen peroxide. It's a toxic chemical, but fortunately, hydrogen peroxide can be neutralized by peroxisomes. Vesicles within the cells are intended, for the most part, for the transport, while vacuoles serve usually as a storage organelle. Vesicles can transport substances because their membranes can fuse with other cellular membranes and can basically unload their content, let's say from vesicles to, from endoplasmic reticulum to Golgi complex. Some cellular structures are unique for animal cells. One of them is a centrosome, which is a center that organizes microtubules near the animal cell nuclei. Centrosome consists of two centrioles. The centrioles are perpendicular to each other and are essentially the cylinders of the triplets of microtubules. 9 triplets form a centriole. Centrioles replicate before a cell divides. The idea is that centrioles may be required for pulling the chromosomes that were duplicated to the opposite ends of the dividing cells during the anaphase of mitosis. Another structure that is unique for animal cells is a lysosome. A lysosome contains digestive enzymes and acid that help to break down macromolecules and recycle organelles. It clearly demonstrates the advantage of compartmentalization. Lysosome contains very aggressive chemicals that the other parts of the cell aren't exposed to because they all are contained within the lysosomal membrane. Just like some structures are unique for animal cells, there are structures that are unique for plant cells. First and foremost, of course, it is the chloroplast. It's the organelle that contains the pigment chlorophyll, capable of capturing the light energy and converting, and eventually chloroplast can convert it into the chemical compounds. The light energy drives the process of photosynthesis, in which carbon dioxide and water are converted into carbohydrates and produce oxygen as the waste product. Just like mitochondria, chloroplasts are endosymbionts. Here you can see the proto-eukaryote acquiring a photosynthetic bacterium that later becomes a chloroplast. Just like mitochondria, they have DNA, they have ribosomes, and they are surrounded by outer and inner membranes. Within the inner membranes, there is a set of thylakoids, stacked membrane sacs, and the stacks form grana, which is plural. Granum is singular, so this is a single granum. The fluid called stroma surrounds the grana, and this grana and stroma are the main sites for the reactions of photosynthesis. Plant cells are also surrounded by a rigid cell wall. It protects the cell from lysis, provides structural support, and essentially warrants the shape of the cell. Cell wall consists of a polysaccharide cellulose, which is comprised by multiple glucose units. You can see the cellulose formula right here. In this image on the right, you can see the cell walls highlighted by the green color. Central vacuole within the plant cell is usually filled with sap. The function of the central vacuole is to maintain the pressure against the cell wall, which allows the cell to maintain its structure. Eukaryotic cells, unlike prokaryotes, have cytoskeleton, which is a lattice of protein fibers. The main function of cytoskeleton in general is to maintain the cell shape, but also to tether organelles to their specific positions, to facilitate the movement within the cell, to give cells the capacity to move. The narrowest types of cytoskeleton filaments are microfilaments, shown here. Microfilaments consist of two strands of the protein called actin, organized in chains. Microfilaments facilitate the intracellular movement and the movement of the cell itself. They are also actively participating in the cell division and the streaming of cytoplasm, directing of the cytoplasm flow. They do provide the rigidity and the shape of the cell, but what's important, microfilaments can quickly disassemble and quickly reform, which makes them quite dynamic, the function essential for the movement and cell division function. Intermediate filaments are made out of several strands of fibrous proteins, such as keratin, that are wound together. Their main function is to maintain the cell shape, to withstand the tension that can be applied to the cell, and to anchor the nucleus and organelles. Intermediate filaments can be composed of different types of proteins, but the keratin is probably the most well known. It can be found in hair, nails, and the epidermis of the skin. The third type of filaments that comprise cytoskeleton are microtubules. Microtubules are hollow tubes that are formed by the dimers of alpha and beta tubulin proteins. Microtubules can resist compression. They facilitate the movement of vesicles within the cell, and they are actively pulling replicated chromosomes to opposite ends of the cell during the anaphase of mitosis. And just like microfilaments, but not intermediate filaments, they can disassemble and reform quickly. I usually compare microtubules and microfilaments to the kitchen cabinets. You can remove the old ones and put the new ones in. Intermediate filaments, on the other hand, can be best represented by the studs and beams of the house. If those are removed, the cell collapses. In addition to their structural and movement function, microtubules also form flagella and cilia, the long structures that make cell move. Flagella can be numerous, or it can be only one flagellum. Cilia are usually in huge numbers. These are short, hair-like structures that either can move the cell or a substance, or they can move substances along the cell surface. So both flagella and cilia have a similar structural organization of microtubules. It's called 9+2 array. A ring of 9 microtubule doubles surround a single microtubule double in the center of the ring. Eukaryotic cells are connected by the extracellular matrix. It consists of proteins such as collagen, the most abundant protein in the extracellular matrix. The collagen fibers form an interwoven structure with a bunch of proteoglycans, and such as this proteoglycan complex and various carbohydrates. Extracellular matrix not only holds cells together, but also allows cells to communicate. Cells may have receptor proteins in the plasma membranes. When a molecule called ligand binds to the receptor, that changes the shape of the receptor. Change in the receptor shape alters the conformation of microfilaments on the cytoplasmic side of the membrane. This change in the microfilament shape may induce chemical signals inside of the cell. These chemical signals reach the nucleus and alter gene expression. If you remember our analogy of the cookbook, when you change the gene expression, you essentially change the dishes that you cook. So that affects the production of proteins. Proteins carry out the greatest number of cellular functions. So when gene expression is altered and the protein synthesis is changed, that changes the activities within the cell. Cells often need to communicate, and they can communicate with each other, often via direct contact, through the intercellular junctions. In plant cells, plasma membranes generally cannot touch each other because they are surrounded by the rigid cell wall. However, the openings in the cell wall, called plasmodesmata, plasmodesma is the singular form of that word, connect adjacent cells, allowing the exchange of materials between the cells, between the cytoplasms of those cells. Essentially, plasmodesmata enable the transport of various materials from cell to cell and, at the end, throughout the plant. In animal cells, cells communicate through gap junctions. Functionally, they are similar to plasmodesmata, but their structure is slightly different. Gap junctions allow the transport of ions, nutrients, and other substances between the cells. They consist of the 6 proteins, called connexins, that arrange themselves in the connexon. Connexin in one cell binds to a connexin in another cell, forming a pore. These pores allow the communication between the cells and help them to establish what's called a functional syncytium. Functional, well, you know the meaning of that word, and syncytium, syn together, cytocells, cells acting together. This functional syncytium is critical for the function of cardiac and smooth muscles, in which cells often have to contract simultaneously. Gap junctions enable a very quick communication between those cells. Not only cells have to communicate, they have to be held together. They can be held together by tight junctions or desmosomes. A tight junction is a pretty tight seal between two adjacent plasma membranes. These seals are made out of proteins claudins and occludins. It prevents materials from uncontrollably leaking between the cells and are typically found in epithelial tissues. Tight junctions, of course, are not like watertight, so there is some movement of chemicals between the cells, but it is, again, very much limited. Desmosomes, on the other hand, simply hold cells together in animal epithelial layers. Proteins called cadherins that are found in the membranes connect to intermediate filaments within the cell. This arrangement allows to maintain cells in the epithelium in a sheet-like formation that is also stretchable. That concludes our conversation about the cells. Next time, we're going to talk about the membranes and the transport.